of aging group, I actually thought that, okay, I know a little bit of bioinformatics, and I felt a bit guilty that I don't use it in an everyday life. You know, it's like being a shoemaker, but not having any shoes on you. And this problem puzzled me a lot. And eventually I decided to sort it out. So first step I did was to sequence myself. And as probably at least 27% of you know, it's very cheap now. So probably the cheapest solutions are Nebula and Dante. You can get 30x coverage, full genomic sequencing with Illumina for around $300 to $500. Many people heard about other services. Probably everybody heard about 23andMe, but it's not a proper full genome sequencing. It's just sequencing some spots in the genome, like 650,000 variants. If you don't have those variants, you don't. So you can only see the variants that they have in a chip. And approximately when you sequence with proper like full genomic sequencing, you get at least 4 million variants. So you got way less than you typically get from sequencing. And there are other also services like Ancestry and self-decode that actually, in my opinion, now provides one of the best health and genetic recommendation. And the second thing that I did, because I was doing a lot of bioinformatic pipelines, okay, I got my sequencing, and let's get to the very raw data. Let's apply the latest tools. And I started publishing this code to GitHub in an open source way. And at some point, I discovered that one anonymous guy with a new nickname Newton Winter with a rabbit on the user picture started to contributing to my code. And from this point, actually, it turned from just a personal project into something something more serious. And now I give awards to Newton Winter. So please follow the white rabbit.
Hello, my name is Newton Winter, and I would like to share history. At the moment, I found DNA-seq repository. I already had my genome sequenced. I got a whole genome sequence. And I had raw reads, but I was not satisfied with reports the commercial service provided, because these were not transparent. They didn't share how did they came to conclusions they provided based on my genetic data. And I also had raw reads on my hands, so it had a potential for usability. So I decided to use an open source pipeline I found, and that's how I joined the project, because I started using and contributing right away. So the sequencing pipeline consists of assembling raw reads against a reference sequence. Commercial services that are widely available provide reads with average length like 100 to 150 bases. It's so-called short reads, and after these are aligned to a reference sequence, variant discovery is done, and those variants are then analyzed and turned into human-readable reports and conclusions. So assembling a genome from scratch is a really difficult task, and it's not only computationally hard, it requires a lot of data. Thankfully, this was done in Human Genome Project that done de novo assembly of human genome. And thanks to that project, we have a reference sequence. By the way, commercial services usually use outdated versions, like previous builds of human genome, and this was exactly my case here. So when you have your raw reads, like a shredded piece of papers, you can align them to a reference sequence of human genome that is also openly provided and available for downloading and usage. And this is done by commonly known Burroughs-Wheeler aligner algorithm. DNA-seq pipeline chose to somewhat shrink amount of job done within the pipeline because technologies develop rapidly, and there is a rather new variant calling algorithm called dict variant that was introduced into the pipeline, and after comparison with other algorithms, it turned out to be the best. It is algorithm that is done by Google. It's based on deep learning, and it's a really clever idea. I'd say neural networks are really good at analyzing images nowadays. So what they did, they basically made a representation of short reads alignments as images, and the convolutional neural network showed its excellence. This algorithm showed like 99.9587% accuracy, absolutely acing the test. And it also excels in solving difficult cases of variant calling when algorithm needs to decide whether the variant exists, or it's a reader, or it's a heterozygous variant, etc., etc. So when your genetic data is assembled into genome and variants are called, there are numerous of types of changes that can differentiate your genome from a reference genome. Changes can happen anywhere. Mutations can happen anywhere in coding regions, in non-coding regions, in promoter regions. So when a change of the base results in change of genetic sequence, but the protein sequence that's produced upon it does not change due to genetic code redundancy, such changes are called silent. Mutation that results in amino acid substitution is called a missense mutation. There is also a nonsense mutation kind when the codon for amino acid is replaced with stop codon, so the protein turns out truncated, and this usually leads to some kind of deficiency, or this protein doesn't function properly. There is also a more severe change, so-called frame shift mutation. It is when genetic code is either inserted or deleted. When it happens within a coding region, it results in a severe change because the resulting protein will turn out entirely different from what it was initially. And this is usually harmful, but not always the case. So deep variant is a very sophisticated algorithm, and making it was only possible because of large amounts of data collected from sequencing. These algorithms different Wheeler's alignment variation we use, namely Wheeler's aligner MEM2, which is somewhat hungry for memory, but it works twice faster. These are containerized Docker containers I used. That allows for easy upgrade, namely different got to version 1.3, and it improved it also already perfect accuracy even further. So as BVA MEM2, it's also somewhat improved, and using containers allowed us to quickly and painlessly updating these parts of the pipeline without need for keeping the environment clean. All this is available on GitHub. You can look at it for yourself, and here's a link. And I believe now I give my microphone back to Anton. Thank you.
Yeah, so with help of Newton Winter, we reasonably quickly got bioinformatic pipelines done. So we got from raw FASTQ with the latest reference genome, not old one like in Dante, to VCF produced by Deep Variant. And the next steps was actually annotating the variants because you have to get meaning out of which mutation. Originally, we used web that some of you may know from Ensemble project, variant effect predictor. Then we switched to OpenCravat, which is much more convenient to use because it has an app store and many databases are represented as applications there. The problem is that when you start working with this, you see like huge number of different annotators, and we didn't know much by the time what to do with them. So of course, the most obvious solution, take all of them. And we selected all annotators, and we start with the second problem, is that after you annotated everything, you imagine you have like 4 million SNPs and well, maybe several thousands. Actually here it's 4,700,000 SNPs, and you get tens of thousands of annotations, and you don't really have time to read all of this. And at this point, we actually got stuck a little bit, and we were continuing in background mode until GitCoin grad round happened. So a friend of mine, Lawrence Ion, told me that there is an opportunity for longevity open source projects to get some cryptocurrency funding. And I was thinking what to apply for, and it was extremely short notice. So I just applied within, I think, 1 or 2 days. So I remembered that I had an idea to actually do longevity annotator, because what I really lacked in self-decode and in Dante reports is information about variants associated with living longer. And also I somewhat lacked more transparent and more serious polygenic reporting. So it was just within just 1 day, we had to take a logo and make a name of the project, and I made very original name, just-dna-seq. And also as a logo, I took 3D structure of DNA from PyMol. With Newton Winter, we expected originally that maybe we can get $1,000 or $2 so we can write a plugin. We also added Olga Borisova to the team because we needed really experienced medical geneticist, and we were really amazed that actually with matching, so the way how GitCoin works is that it's like crowdfunding in crypto, but all funding you get is matched with so-called quadratic formula from the sponsors, and you get much more matching that you get funding, and they try to facilitate small donations. You get higher matching for small donations. And we got like $70,000, and we thought, okay, guys, maybe we should get really serious. So we made a budget for a year and a half, and we increased the number of features we wanted to do substantially. And after we got this funding, I think it was December last year when the campaign happened, and the end of January when we got it.